Hello everyone, welcome. I'm Fuchs and this is the very first video of the Maiolnir channel. I hope that you all survive the disaster that this is going to be, but hopefully we can improve over time. In this presentation, I'd like to answer this question. Can Llama read medical reports? I've been working on this challenge for the past few weeks. Many startups are also trying to solve this challenge, so I'll advance quickly in a hurried manner in the hopes of making this video simple, concise, and going straight to the point. So let's dive in. Here's the challenge. We're going to be receiving medical reports in the format of PDF files. I've created one for us that we're going to be using as a reference for this presentation. It contains the standard information that we usually find on medical reports, information about the patient, and then everything that the doctor has requested for that patient. And given medical reports, we want to have a system that automatically can extract every single biomarker from that report, information associated with the biomarker, and also we're going to have to find an identification of the biomarker which is available on our own internal database. That's the challenge. Okay, you can pause the video now and think for yourself, how would you solve this problem? It's actually quite a challenge problem. Here's the solution that I propose for us to work with. We're going to divide this problem into three main steps. The first step is going to be a summarization process so that you can work with less data. Then you're going to cast this information to JSON so that you can manipulate this data. Finally, we're going to have to implement an AI system that is capable of taking the JSONs and find the identification of the biomarkers. The first two steps, we're going to be using Llama 2. Notice that Llama 2 is particularly well designed for this type of problem, as we're going to be working with sensitive data. So Llama 2, for being open source, can help us here. Let's see how the summary goes. The summary is pretty straightforward. We're going to receive the medical report, and then we're going to ask Llama, make a summary of all the biomarkers that you find in this medical report. Let's see how Llama 2 performs here. Alright, let's go to Colab and start processing our PDF file. I'll make this file available down in the description below, so if you want to follow along, you can also use it as a reference. First of all, we need to install Tesseract in order to be able to extract the text, so let's do it. After that, you need to cast our PDF file to images, so Tesseract can read it. And here we're going to extract the first page of our PDF. Alright, let's see what is the result, and here's the result. Let's copy it and send it to Llama 2 to see how it performs. To test Llama 2, I'll use the model that is available on Replicate, and we're going to be experimenting with the 70 billion model. What we're going to do is, let's paste the extracted text from our PDF. Our system prompt, we won't need it. Let's increase the tokens used. And also let's remove the temperature to have more deterministic answers. The prompt is going to be, give an input text, make a summary of all the biomarkers that you find, and the answer should follow this template. We're going to extract the biomarker name, the test name which it may belong to, the value, and the reference ranges. Here's the input text. Let's see how Llama 2 performs here. Alright, so it just finished processing everything. Notice that it succeeded to retrieve most biomarkers, and it's following the template that we requested. We have a summary of all the biomarkers, but notice if you look at the report, we're gonna see that it should have extracted 15 biomarkers, but here it extracted 13. This system is not entirely capable of extracting everything. There is maybe some prompt engineering that we can do to improve that. So let me show you. For instance, it failed to retrieve erythrocytes. So it maybe can help in the prompt sending this information to see if it helps. What I'm going to do is I'm going to tell Llama to make sure to retrieve erythrocytes. Make sure to retrieve all biomarkers from complete blood count, such as erythrocytes. Let's add this information to see what happens to Llama 2 now. Notice that now it succeeded in retrieving erythrocytes. This is more related to prompt engineering, but what I found in this challenge is that prompt engineering actually does not help very much. If you optimize your prompt to work for one blood report, then it stops working for some other blood reports. In order to solve it, I do recommend using OpenAI Evolve's framework. It is a framework that is going to help you on reducing noise when trying to optimize prompt engineering. In my own experience, prompt engineering will not help much. It's going to improve the accuracy of the system by around 5%, but not much more than that. Well, but the conclusion is the Llama 2, the 70 billion model, actually can read to some great extent medical reports. Okay, now coming back to our colab, we cannot run Llama 2 70 billion model. It will be just too expensive. What we're going to do instead is we're going to run Llama CPP locally so that we have a sense of automation and also can experiment with the system in our own runtime. In order to run Llama CPP, we will require a GPU. Let's change our runtime. Let's change our runtime to get support for GPU now. We're gonna have to reprocess everything. Let's process the report all over again. And now we're going to extract every single text from all of our lab report. Final result should be a list of text files containing the text that was extracted. Let's send it to Llama CPP. Now Llama CPP is pretty much Llama 2, but the model weights are quantized. That is, they are represented as integers. And we're going to be using a representation of 8 bits. This makes the model much lighter so that we can run in our own environment with smaller GPUs. But the price that we're going to be paying is that the quality of the answers will drop. Still, let's experiment what Llama CPP is capable of. We're going to use Llama CPP by installing the Python package Llama CPP Python. And you're going to do so by requesting support for GPU. This code will run in 5 minutes, roughly. Now we download Llama CPP model. We're going to be using a 7 billion model and quantized with 8 bits. After downloading the model, let's instantiate it. We can use the Python wrapper that we just installed. Notice that all the layers of the model will be loaded into the GPU. Just like so, we have Llama CPP ready to run. Let's send a request. We're gonna send the same request that we already did for the 70 billion model. And let's send the first page of our medical report. We're gonna open the medical report in the first page. We're gonna read it and send it to Llama CPP, asking it to make a summary of the biomarkers. Let's see how it performs. So it took around 20 seconds. It's a good time. This is probably already too much, but let's see what the results are. Here's the summary. It does succeed to extract some biomarkers. Notice that it also failed to extract erythrocytes. It does succeed to bring the answer following the template that we have requested. But here, it only extracted 8 biomarkers in total, which means its performance is somewhere around 50% maybe. So despite the model being much lighter and faster, the price that we pay is inevitably answers with lower quality. That is the trade-off. So far, we could say that Llama CPP can be used for local development, but it probably cannot be used for production. We still can move on to keep experimenting. We can see how the system performs when summarizing the whole blood report that we have available. Here, what I'm doing is I'm iterating over all the pages in our medical report and sending it to Llama CPP, asking it to make a summary, and then I'm storing the results in this results variable. Alright, it took 1 minute to process everything, on average 25 seconds per page. Let's print some of the result. Let's print the last page. It does a good job. Iron is a biomarker available on the last page. Notice that the test name is wrong. For what follows, the iron test is also not correct for the test name, but everything else it does succeed to extract. And we also have the same problem as before. It cannot extract all the biomarkers, and performance here is around 30%. It's not good. It's good enough so that you can experiment with it in development, but it's certainly not a system that you can use in production. You can also use it to experiment with JSON extraction. Now that we have all the summaries, we can see how the system performs when trying to cast the summary into a JSON. Doing so is straightforward on Llama CPP. All we have to do is to send a request asking to answer in a JSON format, and then we're gonna send in the request this input response format. It's the same as OpenAI, and you're also gonna be sending the schema of the JSON. Here, we're gonna request an array of biomarkers where each item in this array will be an object with two keys, the name of the biomarker and the value. Let's see how Llama CPP performs in this step. Alright, it took almost 2 minutes to extract the JSON. Let's print the result. For the first page, here's what we got. It does a good job. Notice though, we didn't ask for the key reference range, neither the key test. So those are hallucinations. They don't make sense, but for the rest of the JSON, it's actually pretty good. It does retrieve the name of the biomarkers and the values. So it kind of works. We can work with that, and we could also use Llama CPP for development, but again, we cannot use it in production. And it succeeded on casting all the biomarkers to a JSON representation. We can also experiment with what the 70 billion model would return here. To do so, we're going to use the summary that we have available in the results object. Let me print this value. Let me copy it. And then let's go to replicate again. We're going to replace the older prompt with the new one. Given this input text, retrieve a JSON that follows this schema. Also, let's request the LLM to retrieve only the JSON. And what we're gonna see here is that the performance is remarkably good. It succeeds 100%, and it does extract every single biomarker and their value to the correct schema that you have proposed. So the 70 billion model is actually quite good at following this request of casting to JSON. In my own experimentation, it is quite a reliable system in this regard. And notice that all the biomarkers that is sent in the input were successfully mapped. Alright, we worked with the summary and the JSON extraction. Llama 2 can help us here, but the third step is going to be more challenging. We need a way to extract the identification of all the biomarkers available in this JSON. Let's see how to proceed. Okay, so now we need to move to the third step and the most challenging one. We have the JSONs, but now we need to find a way to extract an identification for those JSONs. That is, we need to understand what has been extracted. Otherwise, it's just a name, and we don't fully understand what this name means. That's why we need to find our own identification. How this works? There's one problem. One way that you could think of is, we have our own list of our known biomarkers, but then the LLM may retrieve names that are not the exact match for our own biomarkers. For instance, sometimes the LLM will retrieve erythrocytes, which is correct, but then it may add some other random text with this name. For instance, also known as red blood cells. We don't have control over that. So if you just try to match the strings from our own database to what the LLM has extracted, they won't necessarily match, which means we cannot use this approach to solve this problem. Another issue here is that some biomarkers, they may belong to different test names. Erythrocytes, it belongs to complete blood count, but it also may belong to midstream stain of urine. So when we extract the JSON, we will also need to use this test name information to know what is the correct correspondent in our own internal database. So how to solve this one? You may think about using LLMs, but these will not work. If we try to use any LLM for this challenge, we may have thousands of biomarkers in our own internal database, so the context length of the LLM will explode quite easily, which means we cannot use it here. Okay, another approach which is much better is using embeddings, but they also will not work. So let me show you why. Let's come back to our colab, and now we're going to be working with OpenAI client because it's easier, and you're going to be requesting OpenAI to convert some biomarkers to their embeddings representation, and we're going to see if we can match everything properly. Let's install OpenAI client for Python, and then I'm going to retrieve the OpenAI API key and set it and make it available in my own environment. Notice that we're going to be using some concurrent processing. Now, I won't dive in details of this code. What pretty much it's doing is sending requests to OpenAI to retrieve embeddings. Now, here's a list that I've created of some examples of biomarkers. Let's suppose that we have that available in our own database. Now, let's suppose that the LLM has retrieved from the step 2 in the JSON's data, something like that. We have the biomarker name, and then the LLM decided to dream something a little bit random after the name. For instance, here it retrieved the cancer antigen 27.29, and then the LLM decided to explain that the abbreviation is CA 27.29. This happens in practice, so we need somehow to be ready to process this type of data. Another thing that I want to mention is, notice that we have erythrocytes for red blood cells, but also have erythrocytes for midstream stain of urine. Both biomarkers are distinct despite having the same name. So the way that we're going to make the distinction in our database is to add information in the name explaining what the biomarker means. The correspondent generated text that we are simulating here from the LLM is the LLM retrieved erythrocytes correctly, and then in parentheses it retrieved which test name the biomarker belongs to. The first one is correct, it belongs to complete blood count, and the second one is also correct, it belongs to midstream stain of urine. Let's get all the embeddings for all of our biomarkers and all the text that was dreamed by the LLM. I'll print the first biomarker embedding just to see what is available, and it's a list of random numbers, or apparently random numbers. This list, I think, has more than 1,000 numbers, and it's a numerical representation of our biomarkers, of the text of each biomarker. With the embeddings, we can create an array of those embeddings by taking the dot product. As those embeddings are already normalized, the result is the cosine between each embedding. And if you use argsort, we can see for each biomarker what is the best fit of the text that was dreamed from the LLM. and then you can print the results. Results are actually remarkably good. For instance, this in the first column, we have the text that was dreamed by the LLM, and then the second column, we have the ID that we have to map to. And if you peruse through the list, you will see that this approach of using embeddings to match text actually works remarkably well. It gets everything right for the biomarkers, but there is one problem. Notice what happens for our erythrocytes entries. For the CBC biomarker, it mapped to the correct value, red blood cell. But for the MSU biomarker, it actually got it wrong. It mapped again to red blood cell. So this is the problem with embeddings. They lack refinement. What we concluded here is embeddings does have a potential of working, but we still need somehow to fine-tune it for our own usage. And that's pretty much what we're going to be doing. So let's see how we can solve this problem. We can actually build our own model for doing this mapping. The approach that we're going to take here is we're going to request the GPT models to dream and generate a lot of text related to biomarkers. The idea is so that we can prepare ourselves to pretty much everything that the LLMs can dream. And then for each text that was generated, we're going to also follow along with the ID of that biomarker. And by doing so, we can build our own model. We can train our own model that hopefully can learn to make this mapping for us. This is in essence, we are creating our own embeddings. Here we're going to be training two models, an embedding bag model and a transformers model. Let's see how it goes. Now let's move on with our synthetic data creation. In order to do so, we're going to be interacting with GPT. Here we're going to be using the 16K model. The first request that I'm going to be sending is retrieve a list of biomarkers so that we have our initial database simulation. Let's move on. Here's the list of biomarkers that GPT retrieved to us. Here I'm just going to create an ID for each biomarker. Now what we're going to do is, first of all, let's process possible abbreviations that each biomarker may receive. So we're going to send to GPT, given this biomarker, answer to me what is the answer, a possible list of abbreviations for that biomarker. Notice that we sent one biomarker at a time, so we have everything well organized. We're going to process everything in parallel, so it's much faster. Let's see the first abbreviation for our biomarker. So creatine is also known as CR. The answer from the LLM that you get from the JSON step may contain the words creatine or just CR or creatine and CR. In a way, we're making ourselves ready for the generated text from the LLM and training our model so that it can successfully map to our correct ID. We're also going to process definitions of each biomarker. Let me show you what I mean by that. For creatine, here's a text defining creatine. This is an interesting step to take if the generated text contains many information that contains even a little bit of hallucination. By training our model with this input, we may have a better chance of successfully finding the correct ID. That's why we are aiming for the extraction of the definitions of each biomarker. Then we're going to process synonyms. Here's an example of creatine synonyms. It's also known as CR, blood creatine, serum creatine, renal function marker. Finally, we're going to ask GPT which tests each biomarker may belong to. Finally, creatine can also belong to these tests. After creating all of this information, we pretty much grab the whole thing and save into a training and validation file. Let me show you what the training file looks like. For each row, we will have the biomarker, and then in parentheses, we will have something that the LLM can generate at random. Maybe it's going to generate an abbreviation. Maybe it's going to generate some text explaining what the biomarker is. Regardless of what the input may look like, this dataset may prepare us to process this input information and still be capable of making the correct mapping to the correct ID. Now that we have our synthetic data, let's explore what the embedding bag model is capable of. The embedding bag model is fairly straightforward and quite useful actually. Here's how it works. It's going to receive our text as input, and then for each token in this text, we're going to associate an embedding, which is a vector with random numbers that will eventually be optimized to solve this challenge. Erythrocytes may be associated with its own embedding, the word red with its own embedding, and so on. What we're going to do then is we're going to average all of those embeddings. We're going to do that for every sentence in our input, and then we're going to use this information to forward it into a linear connection neural network. So we're going to have a fully connected final layer, which is going to retrieve us the logits. And we're going to use those logits to train the model to extract the correct ID that you're looking for. Let's implement it on top of PyTorch. To begin with, let's install PyTorch dataset and data loader, or actually let's retrieve it. We're going to use a custom dataset to work with the data. It's a pretty straightforward implementation. We just open the file and we split with the character that separates the text from the label. Let's create our dataset. Finally, what I'm doing here is we're gonna use a basic English tokenizer. We could use any tokenizer here from Llama 2, OpenAI, but this is gonna be simpler. What we're gonna do is we're gonna process all of the tokens in our training dataset. We're gonna apply small transformations on them. Finally, we're gonna use that to build our own vocabulary. Now what we have is a vocab object that is capable of mapping input text to output IDs that identify those tokens. Let's create pipelines for our input text and our labels. The labels are the IDs of our biomarkers in our database. Now we have the data set, but we need a data loader. The data loader will be fairly simple. Its main goal is to cast the input dataset to PyTorch tensors. Main thing to notice here, first of all, we convert our text to their correspondent IDs, and eventually we're going to use those IDs in the embedding bag model. The offsets is just a tensor of integers that tells the model where each sentence starts or ends. And finally, we have our training data loader and the validation training data loader. Here's an example of a result. First tensor are the labels, then the text token IDs, and finally the offsets. And here we have the model. Notice that the model is fairly simple. We only use the embedding bag, and then we use a fully connected layer so that we can connect to our logits. Now what we're going to do is design a training and validation protocol for our model. Finally, we can instantiate this model and we can run it. Let's see how it goes. In the beginning, it doesn't perform very well. This is the accuracy, which means for all of the guesses of IDs in our training or validation datasets, how much is it guessing correctly? By the end of 30 epochs, it's guessing correctly for around 77, 78. Yeah, something around 78%. Now I can install a function that uses the model that was trained. Let's see how it performs. Now I have here some entries that simulate what Llama 2 may bring to us in the JSONs. Let's see if our model is capable of making the correct mapping. And actually it does succeed. All of our entries are correctly mapped. Notice that I've added some random weird text in our model just to see if it will still be capable of finding the correct mapping, and it succeeded. So for the most part, the embedding bag models already can work fairly well, but in practice, we still can do better. And another powerful model that we can use here is the transformers. So let's see how it goes. Finally, the transformer model. What I'm going to do is the input data is going to be a little bit different. For creating the training and validation data set, the difference that I'm going to do here is I will guarantee that the first tokens are associated with the biomarker itself. And then for what follows is anything that the LLM has generated, such as the abbreviations, definitions, and so on. Let me show you what really changes. We will always have as the first tokens the biomarker name, and then for what follows is anything that was dreamed and so on. The reason that I want to have this guarantee is that so when developing the transformer model, we can always grab the correct token of the biomarker, leave it in the training vector, and then associate it with any text that may follow. This guarantees that we are not training random text, but rather we are training the biomarker related text. It's just so that we increase precision. Now for what follows is going to be pretty similar to the embedding bag. I'll also make use of the NLTK package so that we can remove stop words. For creating the vocabulary, it's going to be quite similar. The only difference between the embedding bag model is that we are removing the stop words. The rest is similar. Finally, let's create our data loaders. For creating the data loader, there is some logic going on. First of all, we need to define the context length of the LLM or the transformer. In this case, we're going to be working with 8 tokens. And I've also created a prior tokens. That is how many tokens we will grab from the biomarker and guarantee that's going to be available in the training point for that tensor. We proceed by extracting a token from the biomarker so that it's always available in the training set. If the token IDs that I've extracted from the text is lower or equal to the context length, then we just pad it with zeros and leave it as is, and we create our tensor with this information. If it's bigger, then we guarantee that the token of our biomarker will be available in that tensor, but also we extract some shuffled tokens from the input token IDs. This guarantees that our biomarker will be trained with some other random text, and hopefully the transformer architecture can learn how to make this association between the biomarkers and any random text that may appear afterwards. By doing so, we have our training and validation data loaders. Result is similar to what we also had before. Now let's create our parameters. I'm using just 3 attention layers. We don't have much data, so we do want to be concise here. Otherwise, the overfitting will be fully inevitable. And also, we are using a much lower dimension representation for our embeddings. Creating the model is straightforward. we instantiate a block which uses a multi-head attention layer. The forward is just a feed forward function. Now the transformer is pretty similar to what we know in the literature. The only difference here is that we have a linear model which transforms the embedding dimension to the total amount of biomarkers that we have in our database. And then we take the average so that we can do the final classification. We are using the transformer architecture for classifying, not for generating new tokens. So that's why we need to take this averaging of the tokens dimension, so that the final result is a vector with all of the predictions for each class. Let's instantiate our model and run it and train it. Notice that it also starts with random guesses for the IDs, but as the epochs advances, the model is also capable of learning something. It converted to something around 80%. The model does seem to be superior to the embedding bag. Let's now see the predictions. Here I'm creating the same function as our collating function of before, but I'm removing the shuffling so that results are more stable, and we can use this function in the prediction. Here we are using our model to run the predictions. Now let's see again how the model performs. I have here prepared a list of entries that the model may have to deal with. And as expected, results are correct. So the model performs fairly well. We can see that it also is capable of dealing with random text. Keep in mind here that we are actually taking only the first 8 tokens, because that's the context length of the system. Probably 8 tokens will be more than enough, but this is something that has to be experimented with. That's how we can solve the third step of our challenge. So finally, to conclude this video, as we have seen, we have walked through the 3 steps to interpret medical reports. The summarization, Llama 2 proved to be capable to some extent of performing. Llama 2 also proved that it's capable of retrieving JSONs. We saw that Llama CPP cannot do that. We can use Llama CPP for development, but we cannot use it for production. And finally, the mapping phase, where we add semantic understanding. This has to be done through a more creative approach. No LLM can solve this step. Well, so the question that we were trying to answer in this presentation: can Llama read medical reports? And I would say the answer is no. While it does perform very well, the performance is not entirely good. It does misses a lot of biomarkers, and it does hallucinate a lot as well. So for the most part, the answer would be no, but I would say that the answer is also but yes. There is a maybe yes. There's still many things that you can experiment with. For instance, we can fine-tune the model by collecting a lot of medical reports and teaching the model how to properly interpret them. This is one possibility, but still, I'm not sure if that would work. Another approach that I also think is very interesting and fun is to collect a lot of medical reports. This would be challenging, but still we can collect a lot of medical reports, and then we can train our own Llama model on top of those reports and teaching the LLM what is the answer that we want from each report. If that would work, I don't know, but I think it's actually the most promising and interesting approach to solve this challenge. So for now, Llama 2 can help us, but it cannot solve this challenge for us. And that's pretty much it. I hope that you all survived to the disaster that this video was, but I tried to make it concise, straight to the point. For those of you who want to follow in more detail all of the functions in the collab and so on, I'm gonna make everything available down in the description below. And I hope that at least you survived the very first video of the Maiolnir channel, and I hope that we'll be able to improve over time. That's what I had to present in this video, and I hope to see you in the next mission. Bye bye.